Biological classification. Since the dawn of the civilization, there have been many attempts to classify living organisms. It was done instinctively, not using criteria that were scientific, but born out of a need to use organisms for our own use, for food, shelter, and clothing. Aristotle was the earliest to attempt a more scientific basis of classification. He used simple morphological characters to classify plants into trees, shrubs, and herbs. He also divided animals into two groups, those which had red blood and those that did not. In Linnaeus' time, two-kingdom classification system with Plantae and Animalia kingdoms was developed that included all plants and animals respectively. This system did not distinguish between the eukaryotes and the prokaryotes, the unicellular and multicellular organisms, and the photosynthetic and non-photosynthetic organisms. Classification of organisms into plants and animals was easily done and was easy to understand, but a large number of organisms did not fall into either category. Hence, the two-kingdom classification used for a long time was inadequate. Besides gross morphology, a need was also felt for including other characteristics like cell structure, nature of wall, mode of nutrition, habitat, methods of reproduction, evolutionary relationships, etc. Classification systems for the living organism have hence undergone several changes over the time. Though plants and animal kingdom have been constant under all different systems, the understanding of what groups and what organism be included under these kingdoms have been changing. The number and the nature of other kingdoms have also been understood differently by different scientists over the time. R.H. Whittaker in 1969 proposed a five-kingdom system of classification. The kingdom defined by him were Monera, Protista, Fungi, Plantae, and Animalia. The main criteria for classification used by him included cell structure, body organization, mode of nutrition, reproduction, and phylogenetic relationship. The three-domain system has also been proposed that divides the kingdom Monera into two domains, leaving the remaining eukaryotic kingdom in the third domain and thereby a six-kingdom classification. You will learn more about this system in the higher classes. Let us look at this five-kingdom classification system to understand the issues and the consideration that influence the classification system. Earlier classification system included bacteria, blue-green algae, fungi, mosses, ferns, gymnosperms, and angiosperms under plants. The character that unified the whole kingdom was that all the organisms included had a cell wall in their cells. This placed together groups which widely differed in other characteristics. It brought together the prokaryotic bacteria and the blue-green algae with other groups which were eukaryotic. It also grouped together unicellular organism and the multicellular organism once. Say for example, Chlamydomonas and Spirogyra were placed together under algae. The classification did not differentiate between the heterotrophic group fungi and the autotrophic green plants, though they also showed a characteristic difference in their cell wall composition. The fungi had chitin in their cell walls while the green plants had a cellulosic cell wall. When such characteristics were considered, the fungi were placed in a separate kingdom, kingdom fungi. All prokaryotic organisms were grouped together under kingdom Monera and the unicellular eukaryotic organisms were grouped under kingdom Protista. Kingdom Protista has brought together Chlamydomonas chlorella, earlier placed in algae within plants and both having cell walls, with Paramecium and Amoeba, which were earlier placed in the animal kingdom which lacked the cell walls. It has put together organisms which in earlier classification were placed in different kingdoms. This happened because the criteria for classification changed. This kind of changes will take place in the future too, depending on the improvement in our understanding of characteristic and evolutionary relationship. Over time, an attempt has been made to evolve a classification system which reflects not only the morphological, physiological, and reproductive similarities, but also phylogenetic, that is, based on evolutionary relationships. Kingdom Monera. Bacteria are the sole members of the kingdom Monera. They are the most abundant microorganisms. Bacteria occur almost everywhere. Hundreds of bacteria are present in a handful of soil. They also live in the extreme habitats such as hot springs, deserts, snow and deep oceans where very few other life forms can survive. Many of them live in or on other organisms as parasites. Bacteria are grouped under 4 categories based on their shapes: the spherical coccus, the rod-shaped bacillus, the comma-shaped vibrium, and the spiral spirillum. Though the bacterial structure is very simple, they are very complex in their behavior. Compared to many other organisms, bacteria as a group show most extensive metabolic diversity. Some of the bacteria are autotrophic, that is, they synthesize their own food from inorganic substrates. They may be photosynthetic autotrophic or chemosynthetic autotrophic. But vast majority of bacteria are heterotrophs, that is, they depend on other organisms or on dead organic matter for food. Archaebacteria. These bacteria are special since they live on some of the most harsh habitats such as extremely salty areas, halophiles, hot springs, thermoacidophiles, and marshy areas, methanogens. Archaebacteria differ from the other bacteria in having a different cell wall structure, and this feature is responsible for their survival in extreme conditions. Methanogens are present in the gut of several ruminant animals such as cows and buffaloes, and they are responsible for the production of methane, biogas, from the dung of these animals. Eubacteria. There are thousands of different eubacteria or true bacteria. They are characterized by the presence of rigid cell wall and, if motile, a flagellum. The cyanobacteria, also referred to as a blue-green algae, have chlorophyll A, similar to green plant, and are photosynthetic autotrophs. The cyanobacteria are unicellular, colonial, or filamentous. Freshwater, marine, or terrestrial algae. The colonies are generally surrounded by a gelatinous sheath. They often form blooms in polluted water bodies. Some of these organisms can fix atmospheric nitrogen in specialized cell called heterocyst. Example: nostoc and anemona. Chemosynthetic autotrophic bacteria oxidize various inorganic substances such as nitrates, nitrites, and ammonia and use the released energy for their ATP production. They also play a great role in recycling nutrients like nitrogen, phosphorus, iron, and sulfur. Heterotrophic bacteria are the most abundant in nature. The majority are important decomposers. Many of them have a significant impact on human affairs. They are helpful in making curd from milk, production of antibiotics, fixing nitrogen in the legume roots, etc. Some are pathogen, causing damage to human beings, crops, farm animals, and pets. Cholera, typhoid, tetanus, citrus canker are well-known diseases caused by different bacteria. Bacteria reproduce mainly by fission. Sometimes under unfavorable conditions they produce spores. They also reproduce by a sort of sexual reproduction by adopting a primitive type of DNA transferred from one bacterium to the another. The mycoplasma are organisms that completely lack a cell wall. They are the smallest living cells known and can survive without oxygen. Many mycoplasma are pathogenic in animals and plants. Kingdom Protista. All single-celled eukaryotes are placed under Protista, but the boundaries of this kingdom are not well defined. What may be a photosynthetic protist to one biologist may be a plant to another. In this book, we include chrysophytes, dinoflagellates, euglenoids, slime moths, and protozoans under Protista. Members of Protista are primarily aquatic. This kingdom forms a link with the other dealing with plants, animals, and fungi. Being eukaryotes, the Protistan cell body contains a well-defined nucleus and other membrane-bound organelles. Some have flagella or cilia. Protists reproduce asexually and sexually by a process involving cell fusion and zygote formation. Chrysophytes. This group include diatoms and golden algae desmids. They are found in freshwater as well as marine environments. They are microscopic and float passively in the water current plankton. Most of them are photosynthetic. In diatoms, the cell wall forms two thin overlapping shells which fit together as in a soap box. The walls are embedded with silica and thus the walls are indestructible. Thus, diatoms have left behind a large amount of cell wall deposits in their habitat. This accumulation over billions of years is referred to as diatomaceous earth. Being gritty, this soil is used in polishing, filtration of oil and syrups. Diatoms are also known as chief producers in the ocean. Dinoflagellates. These organisms are mostly marine and photosynthetic. They appear yellow, green, brown, blue, or red, depending on the main pigment present in their cells. The cell wall has a stiff cellulosic plate on the outer surface. Most of them have two flagella. One lies longitudinally and other transversely in a furrow between the wall plates. Very often, red dinoflagellates, example goniolacs, undergo such rapid multiplication that they make the sea appear red and cause red tides. Toxins are released by such large number of dinoflagellate and that may even kill other marine animals such as fishes. Euglenoids. Majority of them are freshwater organisms found in stagnant water. Instead of a cell wall, they have a protein-rich layer called pellicle, which makes their body flexible. They have two flagella, a short and a long one. Though they are photosynthetic in the presence of sunlight, when deprived of sunlight they behave like heterotrophs by predating on other smaller organisms. Interestingly, the pigment of euglenoid are identical to those present in the higher plant, example euglena. Slime molds. Slime molds are saprophytic protists. The body moves along decaying twigs and leaves engulfing organic material. Under suitable conditions, they form an aggregation called plasmodium which may grow and spread over several feet. During unfavorable conditions, the plasmodium differentiates and forms fruiting bodies, bearing spores at their tips. Spores possess true walls. They are extremely resistant and survive for many years under adverse conditions. The spores are dispersed by air currents. Protozoans. All protozoans are heterotrophs and live as predators or parasites. They are believed to be primitive relatives of animals. There are four major groups of protozoans. Amoeboid protozoans. These organisms live in freshwater, seawater, or moist soil. They move and capture their prey by putting out pseudopodia, false feet, as in the amoeba. Marine forms have silica shells on their surface. Some of them, such as entamoeba, are parasites. Flagellated protozoans. The members of this group are either free-living or parasitic. They have flagella. The parasitic form cause disease such as sleeping sickness, example trypanosoma. Ciliated protozoans. These are aquatic, actively moving organisms because of the presence of thousands of cilia. They have a cavity gullet that opens to the outside of the cell surface. The coordinated movement of rows of cilia causes the water ladle with food to be steered into the gullet. Example paramecium. Sporozoans. This includes diverse organisms that have an infectious spore-like stage in the life cycle. The most notorious is Plasmodium, malarial parasite, which causes malaria, a disease which has a staggering effect on human population. Kingdom fungi. The fungi constitute a unique kingdom of heterotrophic organisms. They show a great diversity in morphology and habitat. You must have seen fungi on a moist bread and rotten fruits. The common mushroom you eat and toadstool are also fungi. White spot seen on the mustard are due to parasitic fungus. Some unicellular fungi, example yeast, are used to make bread and beer. Other fungi cause disease in plants and animals. Wheat rust causing Puccinia is an important example. Some are the source of antibiotics, example penicillium. Fungi are cosmopolitan and occur in air, water, soil, and on animals and plants. They prefer to grow in warm and humid places. Have you ever wondered why we keep food in the refrigerator? Yes, it is to prevent food from going bad due to the bacterial or fungal infections. With the exception of yeast, which are unicellular, fungi are filamentous. Their body consists of a long, slender, thread-like structure called hyphae. Network of hyphae is known as mycelium. Some hyphae are continuous tube filled with the multinucleated cytoplasm. These are called cenocytic hyphae. Others have septae or cross walls in their hyphae. The cell walls of the fungi are composed of chitin and polysaccharide. Most fungi are heterotrophic and absorb soluble organic material from dead substrate and hence are called saprophytes. Those that depend on living plants and animals are called parasites. They can also live as symbionts in association with algae as lichens and with roots of higher plant as mycorrhiza. Reproduction in fungi can take place by vegetative means, fragmentation, fission, and budding. Asexual reproduction is by spores called conidia or sporangiospores or zoospores, and sexual reproduction is by oospores, ascospores, and basidiospores. The various spores are produced in distinct structure called fruiting bodies. The sexual cycle involves the following 3 steps. Fusion of protoplasm between 2 motile or non-motile gametes called plasmogamy, fusion of 2 nuclei called karyogamy, and meiosis in zygote resulting in haploid spores. When a fungus reproduces sexually, 2 haploid hyphae of compatible mating types come together and fuse. In some fungi, the fusion of 2 haploid cells immediately result in the diploid cells. However, in other fungi, Asco and Basidiomycetes, an intervening dikaryotic stage N plus N, that is 2 nuclei per cell, occurs. Such a condition is called a dikaryon and the phase is called as dikaryophase of fungus. Later, the parental nuclei fuse and the cell becomes diploid. The fungi form fruiting bodies in which the reduction division occurs, leading to the formation of haploid spores. The morphology of the mycelium, mode of spore formation, and fruiting bodies form the basis for the division of kingdoms into various classes. Phycomycetes. Members of Phycomycetes are found in the aquatic habitat and on decaying wood, in moist and damp places, or as obligate parasites as plants. The mycelium is aseptate and cenocytic. Asexual reproduction takes place by zoospores and or by aplanospore. These spores are endogenously produced in the sporangium. A zygospore is formed by the fusion of 2 gametes. These gametes are similar in morphology, isogamous or dissimilar, anisogamous or ogamous. Some common examples are mucor, rhizopus, the bread mold mentioned earlier, and albugo, the parasitic fungi on mustard. Ascomycetes. Commonly known as sac fungi, the Ascomycetes are mostly multicellular, example penicillium, or rarely unicellular, example yeast, Saccharomyces. They are saprophytic, decomposers, parasitic or coprophilous, that is growing on dung. Mycelium is branched and septate. The asexual spores are conidia produced exogenously on a special mycelium called conidiophores. Conidia on germination produce mycelium. Sexual spores are called ascospores, which are produced endogenously in sac-like asci, singular ascus. These asci are arranged in different types of fruiting bodies called ascocarps. Some examples are aspergillus, claviceps, and neurospora. Neurospora is used extensively in biochemical and genetic work. Many members, like morels and truffles, are edible and are considered delicacies. Basidiomycetes. Commonly known forms of Basidiomycetes are mushrooms, bracket fungi, and puffballs. They grow in soil, on logs and tree stumps, and in living plant bodies as parasites, example rusts and smuts. The mycelium is branched and septate. The asexual spores are generally not found, but vegetative reproduction by fragmentation is common. The sex organs are absent, but plasmogamy is brought about by the fusion of two vegetative or somatic cells of different strains or genotype. The resultant structure is dikaryotic, which ultimately gives rise to basidium. Karyogamy and meiosis take place in the basidium, producing 4 basidiospores. The basidiospores are arranged exogenously produced on the basidium. The basidia are arranged in fruiting bodies called basidiocarps. Some common members are agaricus mushroom, Ustilago smut, and Puccinia rust fungus. Deuteromycetes, commonly known as imperfect fungi, because only the asexual or vegetative phases of these fungi are known. When the sexual form of these fungi were discovered, they were moved into classes they rightly belonged to. It is also possible that the asexual and the vegetative stage have been given one name and placed under Deuteromycetes, and the sexual stage another and placed under another class. Later, when the linkages were established, the fungi were correctly identified and moved out of the Deuteromycetes. Once perfect sexual stage of the members of Deuteromycetes were discovered, they were often moved out to Ascomycetes or Basidiomycetes. The Deuteromycetes reproduce only by asexual spore known as conidia. The mycelium is septate and branched. Some members are saprophytes or parasites, while a large number of them are decomposer of litter and help in mineral cycling. Some examples are Alternaria, Coletotrichum, and Trichoderma. Kingdom Plantae. Kingdom Plantae includes all eukaryotic chlorophyll-containing organisms commonly called plants. A few members are partially heterotrophic, such as the insectivorous plants or parasites. Bladderwort and Venus flytrap are examples of insectivorous plant, and Cuscuta is a parasite. The plant cells have an eukaryotic structure with prominent chloroplast and cell wall mainly made up of cellulose. Life cycle of plants has two distinct phases, the diploid sporophytic and the haploid gametophytic, that alternate with each other. The length of the haploid and diploid phases and whether these phases are free-living or dependent on others vary among different groups in plants. This phenomena is known as alteration of generation. Kingdom Animalia. This kingdom is characterized by heterotrophic eukaryotic organisms that are multicellular and they lack their cell walls. They directly or indirectly depend on plants for food. They digest their food in an internal cavity and store food reserves as glycogen or fat. Their mode of nutrition is holozoic. By ingestion of food, they follow a definite growth pattern and grow into adults that have a definite shape and size. Higher forms show elaborate sensory and neuromotor mechanisms. Most of them are capable of locomotion. The sexual reproduction is by a copulation between male and female, followed by embryological development. Viruses, viroids, prions, and lichens. In the 5 kingdom classification of Whittaker, there is no mention of lichens and some acellular organisms like viruses, viroids, and prions. These are briefly introduced here. All of us who have suffered the ill effects of common cold or flu know what effects viruses can have on us. Even if we do not associate it with our condition, viruses did not find a place in the classification since they are not considered truly living, if we understand living as those organisms that have a cell structure. The viruses are non-cellular organisms that are characterized by having an inner clustering structure outside the living cell. Once they infect a cell, they take over the machinery of the host cell to replicate themselves, killing the host. Would you call viruses living or non-living? The name virus, that means venom or poisonous fluid, was given by Louis Pasteur. Dmitry Ivanovsky, 1892, recognized certain microbes as causal organism of the mosaic disease of tobacco. These were found to be smaller than bacteria because they passed through the bacteria-proof filters. M.W. Bazernik, 1898, demonstrated that the extract of the infected plant of tobacco could cause infection in healthy plants and called the fluid Contagium vivum fluidum. infectious living fluid. W.M. Stanley in 1935 showed that viruses could be crystallized, and crystal consists largely of proteins. They are inert outside their specific host cell. Viruses are obligate parasites. In addition to the proteins, viruses also contain genetic material that could either be RNA or DNA. No virus contains both RNA and DNA. A virus is a nucleoprotein and the genetic material is infectious. In general, viruses that infect plants have single-stranded RNA, and viruses that infect animals have either single-stranded or double-stranded RNA or double-stranded DNA. Bacterial viruses or bacteriophages that are viruses that infect the bacteria are usually double-stranded DNA viruses. The protein coat called capsid, made up of small subunit called capsomeres, protects the nucleic acid. These capsomeres are arranged in helical or polyhedral geometric forms. Viruses cause diseases like mumps, smallpox, herpes, and influenza. AIDS in humans is also caused by a virus. In plants, the symptoms can be mosaic formation, leaf rolling and curling, yellowing and vein clearing, dwarfing and stunted growth. Viroids. In 1971, Theo Diener discovered a new infectious agent that was smaller than viruses and caused potato spindle tuber disease. It was found to be free RNA. It lacked the protein coat that is found in viruses, hence the name viroid. The RNA of the viroid was of low molecular weight. Prions. In modern medicine, certain infectious neurological diseases were found to be transmitted by agent consisting of abnormally folded proteins. The agent was similar in size to viruses. These agents were called prions. The most notable diseases caused by prions are bovine spongiform encephalopathy, commonly called mad cow disease in cattle, and its analogous variant Creutzfeldt-Jakob disease in humans. CR Jakob जो भी है. Lichens. Lichens are symbiotic association, that is mutually useful association between algae and fungi. The algal component is known as phycobiont and the fungal component as mycobiont, which are autotrophic and heterotrophic respectively. Algae prepare food for fungi and fungi provide shelter and absorb mineral nutrient and water for its partner. So close is the association that if one saw a lichen in nature, one would never imagine that they had two different organisms within them. Lichen are very good pollution indicators. They do not grow in the polluted areas.